So my name is James Reeve, and until about a month ago, I was a PhD at the University of Gothenburg. Now I'm unemployed. So, what I'll actually present today is one of the chapters of my thesis, and this is probably one I'm the most excited about, what I found most interesting. Before I go into details, just huge thanks to the people that helped me out, especially my supervisors, Kirsten Johannesson and Roger Butlin, who's in the audience. And I'm looking for a postdoc, so if you like what you see, let's have a chat. What I was looking at is ecotypes. And if you're unfamiliar, ecotypes are ecological units that have inherited phenotypic differences that arise from genetic responses to particular habitats. So you go from kind of a neutral ancestral state, and in a very aquatic environment, you develop aquatic features, and in a more dry environment, you develop different features. But I didn't do the research on Pokemon. I did the research on a sea snail that you find in the Atlantic called Lidorhinus saxatilis. And past research has heavily focused on two ecotypes that you see along this transition zone between boulder fields, where you get this larger snail with thick shell that's adapted to resist predation from shore crabs. And then on these rocky headlands, you get this wave ecotype, which has a much smaller size, but the opening area of the foot is much larger relative to the body size. And this allows it to attach onto the substrate and avoid getting washed away in storms. So that's kind of a repeat. So we have these habitats, but then there's other habitats too where we find these snails. We have these, for example, marshy areas that are very sheltered, and we don't really know what is there. Is it a crab ecotype? Is it a wave ecotype? Is it something else? So for my PhD, what I did is, during the COVID summer, go out to visit 200 randomly chosen sites and collect snails along a transect. So at the bottom you can see that photo, and you might be able to see the yellow squares. We laid them out along an 8-meter rope that went from the lowest point where we found snails to the highest point, and each meter we collected the snail we found right underneath the rope. You can see above that just the sheer range of different phenotypes we found. This is only 9. I collected 1,488 snails, so this is the exceptions or the interesting ones. And then we also looked at a lot of different environments, and you can see some of the different shorelines in the photos that go at the map. Then I also took genetic data from the foot tissue of each snail. So I have environmental data, phenotypic data, and genetic data for all these snails. So focusing on the phenotypes, I did a PCA to rejig the dimensions and get kind of the key phenotypic variants. So here you can see kind of the dots spreading out, and if you had really discrete ecotypes that had strong phenotypic differences, you'd expect clusters. But instead, it's a cloud of variation, and when we infer the ecotypes based on the shell photos and the environmental photos, you kind of see two different sides of this cloud of variation. So what we're looking at is a phenotypic continuum, and the ecotypes are kind of the edges of this continuum. Then also, you'll notice a lot of those outliers where I've kind of drawn the lines out to. They're undefined. I realize the colors aren't so great on this screen, but they're kind of little gray dots. They don't really fit one of the two ecotypes. So there's also a lot more things going on here. Then to kind of bring in the environmental data, I did a similar kind of PCA-based approach on the environmental scores, and these plots show the comparison. So environmental variance dimension 1 is that first primary axis, and then you go down to dimension 2 and dimension 3. And the y-axis on all of these are the phenotypic axis. So you can see up the top, there does seem to be a strong correlation with ecotype. The wave ecotypes all on the left-hand side, so your right-hand side, and the crab ecotypes all on the left-hand side. And this dimension, when I looked at the factor loadings, seems to be really related to kind of the wave exposure and how much wave force there is at the shore. But then the other environmental dimensions don't really have a strong association to these ecotypes. But then when I kind of just look at the overall associations in the tile plots here, so the color represents the effect size, basically the slope of the relationship. And if it's completely white, and you might be able to just see the faint lines running through it, that indicates it's a non-significant association. So the really bright or really shaded areas are really strong associations, and a lot of different phenotypes are corresponding to different environments. This gives me a sense that there's a lot of different aspects of either plasticity or adaptation going on that are linking variance in the environment to variance in phenotypes. Then did the same with the genotypes. So here I'm focusing on chromosomal inversions that we found in the genome, and the 5 separate box whisker plots. Just as examples, there's a lot more sites that I looked at. And for some, like the bottom left one, 9.1, you don't really see much of a difference. And then for others, like the top right, 6.1.2, you actually see quite a lot of difference. And anyone that's curious, there's 6 genotypes because this is a complex inversion. You basically have an inversion and then an inversion on top of that. And for 6.12, in particular, we see a really strong correlation with the crab ecotype and one particular genotype of the inversion. So this is a previous candidate we've seen in other studies, and it keeps popping up as this strong ecotype determinant. But again, when I do the tile plot, you see a lot more out there. There's a lot of, if you just focus on each of the rows, most of them have an association with some phenotype, which indicates that it's not just ecotype determination. There's a lot of different effects going on in this whole landscape. So what I think is going on, like as an overall picture, what I think is happening is we have a complex landscape where there's a patchwork of different ecotypes. You've got the orange parts where you have, and blue parts where you have these really strong, clear ecotype differences that are met in these hybrid zones. Then on the backside of the island, you have more of a continuum, or you just have something very different, like very sheltered, marshy areas. And I would call these kind of the intermediate populations. So in conclusion, what I show is ecotypes are part of a continuum, especially if we're looking at phenotypic data. And we have these intermediate populations outside of the major habitat transitions, where you can get very independent adaptive effects. But this said, I'm only looking at one species here, and I think it's really important to investigate this across, investigate the ecotype question across a whole range of different species, and using this landscape sampling. Don't just look at hybrid zones. Sample more randomly. Try and get the whole spectrum of local diversity. So with that, thanks for listening, and we have 2 minutes for questions. Actually, quite a bit of time for questions.
How much movement do you see of these snails during their lifetime, like over one of these 8-meter transects or around the island at all? Are areas where there is a really strong ecotype difference along a transect able to move up and down that freely, or are they kind of relegated to one habitat or the other?
So the habitat transitions tend to be like 30 meters or so, and our estimates in the past have been about 3 meters of movement in a total generation. So it can be really limited, and I think it is a case that we actually do have quite not so much gene flow kind of going on and not so much dispersal across these hybrid zones, but there is a lot of something we haven't really investigated is long distance dispersal, and can you kind of, for instance, get a snail that's knocked off the shore, winds up somewhere else, and then starts to introgress and hybridize. Yep.
Do we know how old the inversions are?
So the question is, do we know how old the inversions are? Not really. I can't give you like a specific date in millions of years, but we do have several other species in the genus that have these inversion polymorphisms. And based on kind of the estimated divergence times, probably somewhere, if I hazard a guess, somewhere from like 10 million to as young as like 1 million years. And each inversion is kind of its own story. So this is still very much what we're actively looking into. Yep.
That is very cool. Do you get the sense that each like phenotype or the intermediate phenotypes are getting like some fitness optimum in different environments, or could they be like come up by chance and they like happen to survive, but they're not meaningfully contributing to subsequent generations?
Yeah, that's a really good question. And so it's kind of the question was, do the intermediate populations have some independent direction of selection, or is it kind of spurious and by drift? And I would guess probably a bit of both, but we haven't really investigated these relationships at like a specific site level to look at what are the main drivers in each site. And just anecdotally, some of the sampling areas seem to be population sinks. We found a lot of dead snails and empty shells around the area, and I have a feeling, even though we were sampling the live ones, it was probably they're just getting washed up and dying within a couple of years or so. Yep, go ahead.
So, remind me, where do you find Saxatilis? Do you find it in the Kattegat, but do you also find it in Norway?
So Saxatilis can basically be found in the Kattegat out to like Bornholm, and in Norway all the way up to North Cape.
Right, because there's quite a striking tidal difference between this side of Norway and this side of Norway, and I was thinking, if has any work been done on sites that have a much more striking tidal difference, like on this side of Norway versus the Kattegat, that it can be sort of fickle related to the weather. It mostly drives the splash areas. Do we know anything about this type of sort of the intermediate, the crab and the wave, as a function of sites that have a much more sort of defined intertidal gradient?
So currently no, but we do have the data to look into that. So we've got, for example, data from the English Channel near Roscoff, where you have the tidal difference like the Bay of Fundy. You can walk out to islands in the middle of the day, or at low tide, and that will be really interesting to look at if we do a similar kind of landscape-based survey and collect these intermediate populations to just see if we do have this sharper difference because of the stronger tidal change. Thanks.
All right, volume good in the back, thumbs up in the back. Okay. Hi everyone, my name is Sam Mantel. I'm a postdoc at Purdue University in the Oakley lab, and today I'm going to be talking to you about some work that I've been doing, trying to investigate the genetic basis of a large effect trade-off across environments in locally adapted populations. Oh, should have checked this. There we go. All right. So to start off, I'm primarily interested in thinking about this question. What's the genetic basis of fitness trade-offs driving local adaptation and biological diversification across environments? And I'm going to be defining local adaptation based on a native versus non-native fitness. So in this example, we've got two populations or ecotypes that each have a higher mean fitness in their home site than they do at their non-native site. And this has been studied quite extensively from classic experiments like Klausen-Keck and Heisey all the way through today that have shown that local adaptation is quite common but not ubiquitous. But when it does occur, we have comparatively little information known about the mechanisms underlying that adaptation, primarily what traits are important and what genes are underlying those traits. And here I'm proposing that Arabidopsis thaliana is a great system to study this type of thing for all of the kind of general reasons that you all already know: genetic and genomic resources, highly fecund, time and space efficient, but more importantly for this, because of their large native range with natural populations that are adapted to a wide variety of different environmental conditions and very little gene flow between those populations. So here I'm showing you a map of Western Europe with Thaliana's Eurasian range shown in gray. And I'm marking two populations, one in Sweden and one in Italy. And as you can imagine, these two populations experience very different climates. So at the Swedish population, plants are living as winter annuals. They're germinating in late summer or fall, and then have this long overwintering period where we have strong selection for survival during this harsh, cold winter. And then they are flowering and producing seeds in the spring. At the Italian site, likewise, they're also living as winter annuals, but they have a much shorter lifespan here, germinating much later, overwintering for a much shorter period, and then setting seeds earlier in the spring before terminal drought ends their growing season in the summer. So then in Italy, we have pretty strong selection for earlier flowering to avoid that terminal drought, essentially. And we can look at this seasonal variation in another way here, looking at maximum and minimum temperatures kind of across the growing season at both of these sites. So as I showed you before, the growing season at the Italian site is much shorter, but a couple things that I want to specifically point out, one of which is this very harsh winter in Sweden where we routinely have below freezing temperatures that these plants are going to have to survive. That does not occur at the Italian site. But also more importantly, early on at the Swedish site, we have these cooling temperatures that have not yet hit freezing, but they're going to allow the Swedish ecotype to sense that winter is coming and acclimate to those freezing temperatures. And what you can see down here in the Italian site is that we still have these same slightly low temperatures between 0 and 10 degrees Celsius that the Swedish ecotype is able to use to acclimate, but that's existing kind of consistently across the entire season in Italy. So, as you might expect, when we do a reciprocal transplant between these two sites, we see a very turned graph that was not the way it looked when I downloaded this. So throw your head sideways. The takeaway is the same. The reaction norms are crossing. There's local adaptation. You'll have to believe me there. All right. This is based off of 8 years of field data from reciprocal transplants done at these sites. And I was trying to show you relative fitness differences. It's a very nice, clean graph, if you can see it correctly. But based on what I was saying on the last graph, I wanted to point out that this local adaptation is due to many different loci kind of across the genome that have many different effects in many different directions. But a couple things that I want to point out, and I don't know if they'll pop up in the right spot, but winter mortality is leading to the Italian genotype doing poorly in Sweden, as we might expect. They're not acclimating to these winter temperatures and then dying during the long, harsh, cold winter. And my other thing didn't pop up. Oh, reduced fecundity, maybe, right there. Maybe less obvious is that in the Italian environment, the Swedish ecotype, like I said, is still experiencing these somewhat low temperatures that allow them to begin to acclimate, but then never experience the freezing stress that they're expecting, and that is leading to reduced fecundity in these populations. So those are kind of the two main things I want you to think about in terms of the local adaptation and a potential trade-off in this system. So here I'm showing you the results of 2 QTL mapping experiments using a real population that was made between these ecotypes. So one is showing average fitness over those same 8 years in the field, and those in the red and the blue arrows. But the other is mapping freezing tolerance in the lab, and those are the black arrows. There's a lot going on here, so I'm going to zoom in to just the 4th and the 5th chromosomes to show you the loci that we're kind of most interested in. So one of them is right down here, and that's just to orient you, the directionality of these arrows is showing the effect of the Swedish allele specifically. So down here we're seeing a locus where the Swedish allele decreases fitness in Italy, increases fitness in Sweden, and increases freezing tolerance. And these are all co-localizing. So this is the kind of genetic trade-off that I would be interested in looking into. We can hypothesize that there might be one single locus that's leading to all of these changes. So we want to understand and try and fine map what the genetic basis of this trade-off is happening here at the very end of chromosome 5. And then briefly, I'll also show you here on chromosome 4 another locus that shows that same pattern that has already been identified by the lab. We have this gene CBF2 where we've identified the causal mutation that leads to this trade-off. You can read about that more at that paper there if you're interested. So we want to fine map this one at the end of chromosome 5. One of the things that is really powerful about this system is that we have this nil or near isogenic line panel in the Swedish genetic background that will allow us to narrow in just to this region of interest at the end of chromosome 5. And if we zoom in there, we can see that we have these 4 different nils that all have overlapping introgression regions right at the end of chromosome 5, but that aren't identical that we can use to try and fine map this region. And we can do that by looking at these nils and trying to measure their freezing tolerance. So that's what I'm showing you here. We took those 4 nils and both of the parental ecotypes and did a freezing tolerance assay where we grew the seedlings at 4 degrees Celsius for 10 days to allow them to acclimate, and then challenged them with a -7 degrees Celsius freeze for 7 days and measured their survival. And as you can see here, we have a roughly 20% decrease in survival through this freeze compared to that Swedish ecotype across all of our nils pretty consistently. So that leads us to believe that our causal locus is in this small overlapping region among these 4 nils that we have. And that encompasses about 670 kb, which still has 185 genes, so still a lot to go through. We'd like to try and narrow that list down even more. And one of the ways that we can do that is to look for cold inducibility. So, let me see here. Is it switching now? And we're frozen. Technical stuff, awesome. All right, if I click the mouse now, it works. So we're looking specifically for cold responsive genes in this region of interest now, because we should expect that genes that change their expression during cold acclimation are going to be our most likely candidates. We're expecting Sweden to be doing something when it's acclimating, up or down regulating particular genes that Italy ecotypes might not be doing. So to do that, we grew seedlings in 2 different conditions, the Italy and Swedish ecotypes, along with 2 of those nils that we looked at previously. Some of them we grew in what I'm calling a pre-acclimation treatment, so just normal growth at 22 degrees Celsius. Others, post-acclimation treatment, where we did this same 4 degrees Celsius treatment for 10 days to allow any expression changes to manifest. We did mRNA sequencing, read counting, and then did differential gene expression analysis in EDGE-R. So to look at this graphically, we're looking for something kind of similar to this, where these ecotypes are differentially cold responsive, and so they respond to cold acclimation, represented by this higher slope, specifically in the Swedish ecotype from pre-acclimation to post-acclimation treatments. So this Swedish allele in this example would be more cold responsive than the Italian allele. And just to reiterate, we're doing this looking for cold responsiveness, which is a significant interaction between genotype and cold acclimation treatment on gene expression. So this contrast that I'm showing here is going to be kind of our first pass. We should know that between these 2 ecotypes, we should see different cold responsiveness in Sweden and Italy. But we can be extra conservative and look at extra contrast to try and minimize type 1 error rates here by looking both at the comparison between the Swedish and the Italian ecotype and also between the Swedish and both of these nil genotypes that we have. As you might expect, more technical issues. So you can't see any numbers here, but I promise that inside where all three of these contrasts overlap is the number 108, because there's 108 differentially expressed genes that are found in the same direction in all three of these contrasts between Sweden and Italy and between Sweden and both of the nils. This one actually does work, where I'm showing you the locations of those 108 genes across the 5 chromosomes of Arabidopsis. As you can see, we've got a cluster of them right in this overlapping region where the nils were introgressed, as we would expect, but we can also see that there's a bunch of trans effects happening, so there's probably microRNAs, transcription factors, other things that are influencing expression kind of throughout the genome. But ultimately, when we're trying to find map here, we're only interested in the actual genes in that overlapping region. So we start with those 108 shared differentially expressed genes. 30 of them are on chromosome 5, but only 7 are in this overlapping 670 kb region that I showed you previously. And here's a list of those genes. The top one that I've got bolded is MAF3 that I'm going to be most interested in for a couple of reasons. One of those is this. So looking at MAF3 expression, I'm showing you the log2 counts per million for our 2 ecotypes and the 2 nils that we tested, both pre and post acclimation. Very slight 0.7 times increase in expression in the Italian genotype, much more extreme 2.1 time increase in our Swedish ecotype. So this is the kind of directionality and change that we might expect for a good candidate. But the main reason I'm most interested in this MAF3 gene in particular is this one base pair deletion specifically found in the Italian ecotype. So we've got one SNP kind of upstream, but then a deletion of this last terminal A here that creates a frame shift at the 15th amino acid of this protein. So I was pretty happy to find that. Seems like a pretty good candidate. Here's just a summary. MAF3 is MADS affecting flowering 3. A MADS box transcription factor with experimental evidence previously that associated its expression with vernalization, flowering behavior, and abiotic stress. But we haven't seen any previous literature associating this with cold responsiveness or cold acclimation as of yet. We see this small expression change in the nils and in our Italian ecotype, a much more extreme expression change in Sweden, and this maybe, hopefully, very important frame shift mutation in the Italian allele. I'm mostly out of time, so I'm going to go quickly through future directions. I'm going to try and functionally validate this with CRISPR. We can try and knock out the MAF3 allele in the Swedish ecotype and see if that decreases freezing tolerance and decreases mean fitness in growth chambers that are mimicking the Swedish environment. Vice versa, with adding a functional Swedish copy to the Italian ecotype. If those things don't pan out, we've got our 6 other candidates that we can keep looking at. And ultimately, we want to look at combinatorial effects with CBF2, our other important trade-off loci that I introduced earlier. I'm done. Thank you.